Hello and welcome to today's Introduction to LabKey Biologics webinar. My name is Kelsey Gibson. I'm going to be your host for today's session. If you experience any technical difficulties with your audio or video, please feel free to reach out to me. I'll be monitoring all of those throughout today's session. We're going to give everyone just a couple of minutes to get logged in before I pass it off to Ryan. But I wanted to let everyone know that we will be doing some questions at the end. So if you have any things that you would like answered that come up throughout today's presentation, go ahead and use the question panel on the GoTo webinar interface. You can add questions there, and then we'll answer those at the end. We'll also take a little bit of a pause once Ryan finishes walking through the platform to submit any additional questions. We're going to get going in just 1 minute. Thanks so much for joining us. We'll get started shortly. All right, at this point, we're going to go ahead and get started. So today's webinar is all about LabKey Biologics. Our product manager, Ryan Luce, is going to walk through kind of the background of the product and some of the core areas of functionality, and then really dive right into taking a look at what it looks like in action. So without further ado, Ryan, go ahead and take it away.
All right. Thank you, Kelsey. It's nice to meet you all. Thank you very much for taking the time to learn a little bit more about LabKey Biologics today. I do want to re-emphasize something Kelsey said. You know, this is a really large application and sort of a product area, and the chances that we're going to miss something that you wanted to hear about are high. So go ahead and just submit questions as they come up, or even if they just kind of occur to you, and we'll make sure to address those once we're done with the sort of demonstration of the application itself. So what we are going to talk about today, first of all, just real briefly, I promise you, a little bit of history about LabKey and the development of LabKey Biologics. Current challenges that we saw within the sort of molecular biology laboratory and what we wanted to address, how we went ahead and sort of built the application, the various components, as well as a demo of LabKey Biologics, and again, that question and answer period at the end of the talk. So LabKey has been around for 14 years. The initial instance of LabKey server was deployed at Fred Hutchinson Cancer Research Center here in Seattle, Washington in 2003. And the company was spun out as a standalone company in 2005. So even though LabKey Biologics is a fairly new product launched earlier this year, it is built on the foundation of LabKey Server, which has been used by hundreds of clients around the world for well over a decade. And really during that time, we've continued to build a variety of different features, integrations, reports within LabKey Server that help research groups manage biomedical research data. Now, specifically around biologics, in 2015, we were approached by Just Biotherapeutics, which is a fairly new biotech company that is also located here in Seattle, to build a new application that leveraged LabKey Server as a foundation, but was really focused on the workflow, data management, and registration of molecular biology entities. So we launched LabKey Biologics earlier this year but continued to actively sort of develop on that platform and that application. So as we worked with Just Bio, and as well as a number of other companies, the question is, what do molecular biology teams need? What do we need to do to enable this team-based science? So the first piece of that, and kind of foundationally, is easy registration of biological entities. And I'll show you how we do that within the system, but the key thing to remember is that the entities themselves are fairly configurable. So we've got, protein sequences, nucleotide sequences, cell lines, constructs, etc. And that we always provide a number of different ways to actually register the entities. The next piece is really that sort of connection between experimental data and the sequence information itself. You need a scientist needs to be able to understand what is the lineage of a particular sample. That gets into the tracking and querying of that lineage, being able to do that in a performant manner so that queries don't take, hours or minutes, but take seconds in terms of understanding what a sample is really constituted of. The system has to be customizable. Obviously, there's a lot of overlap in how research institutions approach biologics, but there is obviously differences, both in their kind of the entities of interest, the vocabularies, and so we needed to make sure that was built into the system. Additionally, tools to manage laboratory workflows. In any of these instances, it's rare to have a single scientist doing all of these bits and pieces. We needed tools to help foster communication, make requests, understand what data had been submitted or not, and so building a workflow management tool was a key component. And then the final piece is integration with external software and instrumentation. So whether that is a specific analytical instrument, being able to upload directly to LabKey the results of a particular experiment, or other software such as existing registration systems, inventory modules, etc. LabKey, because of its history and because of how it was built, is really able, I guess, to build those types of integrations or foster those types of integrations. So more specifically, in terms of how we built this with Just Biotherapeutics, we've been working with them for over 2 years now, and they were really kind of our flagship partner in defining what those requirements were. But we also know that we were building a product for a much broader audience than a single biotech. And so we established an advisory council of pharma and biotech companies to guide the functional and usability needs. So this is a formal product advisory council that we meet with every single month to get feedback on initial designs, what we've built, what our plans are. One of the more valuable things that can be provided to us is actually sample data to make sure it sort of fits into the system. And a number of those early participants in the Product Advisory Council are now customers of LabKey Biologics. We launched version 1 earlier, the beginning of 2017, but we are continuing active development, and the specifics of that roadmap are very much available upon request if folks are interested. So the architecture of this particular application. I mentioned this earlier, but it's really critical to understand that LabKey Biologics is an application that is built on the foundation of LabKey Server. So what LabKey Server is a data integration framework for a variety of different types of biomedical research data, bringing data from different assays, different clinical trials, whatever it may be, all into one place where scientists and clinicians can collaborate and look at sort of a single source of truth of all of the various data that is coming into an institution. It's important, however, then to have a powerful querying engine that allows us to connect those types of data. And one of the key aspects of the LabKey Biologics piece was not only being able to query, but query in a performant manner to be able to get those answers back quickly. We have a robust security model, which means that there is permissioning and roles available for all the various individuals that are using LabKey Server. and then ultimately a developer-friendly architecture. So people can build their own modules, use the existing the APIs that we use in the application itself to build additional functionality that we may have not developed quite yet. So on top of that foundation is LabKey Biologics. And so there are 5 different pieces to LabKey Biologics, and I'll kind of walk through those in it shortly. The first of those, and sort of most critical, is the entity registration piece. Being able to register sequences, molecules, constructs, and cell lines, a variety of different things for tracking those bits and pieces throughout the system. We also have a media registration module. Now, some folks are not necessarily as interested in that, but a number of folks very much are, particularly when we talk about optimizing bioreactor runs and being able to really improve the yield of antibody or protein or whatever it may be that you are developing within the lab. So we track media recipes and media batches if so desired in your particular deployment. Right there in the middle is sample tracking. And so a lot of the key aspect of LabKey Biologics is being able to understand the physical lineage of any particular sample. You know, this particular sample was initially a bioreactor run, then we went through some purification step, and it is a particular fraction from whatever that may be. You know, many generations of basically a sample generation, many generations of samples, if you will. Being able to go back through that and understand, okay, well, what expression system or what cell line did this sample come from is a key component, and that's all sort of automatically done within the application. Integrating all of that sample tracked samples with assays, and then finally that workflow management piece I previously mentioned, being able to see, being able to make requests that assays be done or other sample manipulations be done. It's very configurable. In fact, you could use it to manage, you know, things such as paper preparation or whatever you want. But basically, it's a request system for being able to do that that is tightly integrated with the rest of LabKey Biologics. So let's start. Just before we go into the actual application, the first piece, like I said, and sort of most critical, is entity registration. Now this is, it obviously comes pre-configured with a variety of kind of key entities, but that can be customized for your particular laboratory workflow. Most important, though, is kind of that second big bullet, which is the registration methods. We've built wizards or forms for registering these individually, but oftentimes you need to register things in bulk. You need to upload as quickly as possible 1,000 different amino acid sequences. And LabKey for each of these entities has built-in capabilities to be able to do that. Just as critical, you can register these entities via our API. Now, what that means is that, you know, a different program or a different computer can basically talk to LabKey Biologics and register these things directly without someone actually ever going into the browser and registering them kind of on their own workstation. Also in that entity registration, we obviously have mechanisms to translate sequences, track the lineage of entities, and always ensuring uniqueness in the system, making sure that that protein sequence or that nucleotide sequence or that exact antibody with that heavy chain and light chain has not been registered before. So that's a kind of a key component of the entity registration piece. So let's get out of PowerPoint and actually go look at the application. So this is the you know, the landing page. Oops, got a little cover piece there. There we go. This is the landing page when you log in to LabKey Biologics, and you can see we've tried to make it a very user-friendly and easy to sort of start that navigation piece. We have a large search bar here, and then these 5 different tiles match the 5 different areas that I was just talking about. So specifically, I was talking about the registry. And so pre-configured, we have these different entity types within the system. And I can click on this little guide here, and it'll show me the relationships between some of these entities. So for instance, a sequence, a nucleotide sequence, is inserted into an empty vector to create a construct. A host cell line and a construct are combined, can be combined to form an expression system. You can also actually have a single cell line and multiple constructs that are used to form that expression system. But ultimately, that expression system generates molecules. Now, molecules are a highly flexible sort of concept and can be composed of protein sequences, nucleotide sequences, other molecules, or other things. The demo data that I'll show you later is pretty biased towards antibodies, but the system is very much flexible to allow for a variety of different other types of molecules as well. So I'm going to close that little guide, and I was going to go visit our protein sequences. So this is a grid of the, to this point, 52 different protein sequences that have been registered within the system. And you can see within this grid, we can actually, if we want, we could, for instance, we could search for just cross-mab, and we would see all those that have that, you know, within their chain format. Or we can, for instance, filter a column. We're going to filter, let's say the average mass is greater than 24,000. So there's a variety of different ways we can kind of look at that, and you can, you know, these filters and sorts are additive, so you can add as many as you want within there. And then you can also, you know, download this grid of data directly if you want to bring it into Excel, and this grid actually includes the full sequence. We don't see the full amino acid sequence here, but if I download it, I will absolutely get it in my, oh, I don't have the security permission there. But basically, you can download that full grid of information into your system. That actually was a good example of some of the permissioning has to be set up such that that individual has the right to download those types of information. So let's go look at a particular protein sequence. In this case, we're going to look at PS13. You can see when I sought out PS13, we actually got two. One was the sequence itself, and then another that had PS13 in the description. So when you're doing those types of searches, it's searching across all of the different fields. If I go look at PS13, you can see we have information in the description, nucleotide sequences that encode for it, its chain format, aspects of its physical characteristics, and then down here the sequence itself. And we've got annotations on, you know, the CDR1, the kappa variable, all within the sequence itself. I can also, you know, copy the sequence or parts of the subsequence into my onto my clipboard. And this is a recurring theme with everything in LabKey, is that we try to make, we know that there are some folks that kind of like, we want you to do all the manipulation of data within the application itself. With LabKey, we know that's not really a possibility. You're going to have other analysis tools, other ways of viewing these things, and so we always try to make it as easy as possible to both get data out, in this case, and also to get data in. So those are always kind of options within the system itself. For this particular protein sequence, too, we can also see other entities to which it's connected. So in this case, protein sequence 13 is part of M3, which is an antibody. It has this description, and we can look. It's got two components. PS13, which is the second one, was the light chain. PS14 is the heavy chain, and we can get a little bit more information about those chains right here on this particular view. This particular molecule is seen in this expression system, and you can see how basically all of these entities are linked together once they're registered within the system. Most critically, too, is on this expression system, not only do we see the links to other entities, but the physical samples that were derived from this expression system. So in this case, we have a couple of different sample types, and we can, if I click on either one of these, it's going to take us to a filtered list of all of those various samples tied to that expression system. All right, so I'm going to jump back into my slides here. So we've talked about entity registration. The next piece is sample generation. And one of the key components is that we want to be able to generate samples very easily, being able to talk a bit. One of the surprising aspects of this project, we did not realize this at the beginning, was how important it is to be able to configure how sample IDs are generated. So we make it very easy within the system for each sample type to have different ways of generating that sample ID. So it can have a prefix like S or whatever prefix you want to indicate the sample type. You can incorporate the date. You can incorporate, the lot, other aspects about it, and then sort of integers on top of it, always making sure to make sure each sample ID is completely unique. We also make it very easy to track that lineage. What was the sort of parent of this sample or this group of samples, or pooling multiple samples into a single sample. So I'll show that in just a sec. But the other thing that tracking that sample lineage allows us to do is to go all the way back to the registered entities to which they're connected as well, and view experiment results side by side with the sample, target lineage information about that, so that whenever you're looking at assay results, you can tie it back to the key entities that are connected to that particular sample. You can easily navigate to that detailed information and conduct a variety of different analyses, whether they're visualizations, or you want to integrate with something like R, or export entirely for some sort of other external analysis. All right, so I'm going to go back into the application. And I don't know if before I jumped out, I clicked on this sort of top level sample. So this is a heat map basically indicating activity. You can see I loaded up a lot of this dummy data a couple of days ago during the month of October. And if I go look at any one of those samples, so let's go just look at a particular sample detail page. And you can see over on the right, we've got this really nice lineage of how that sample was generated. And in this case, that it's connected back to expression system 3. We also have a link to the data that is connected to this particular sample as well. But one of the things I wanted to show is if we go and visit the parent of that particular sample, we're going to load the lineage here, and we can see that there's actually 39 different children of this particular sample. So the sample we were looking at had 38 siblings, if you will. We're actually in the midst of developing kind of a full screen view of these types of lineage. We run out of real estate, frankly, very quickly once you start having 10s or 20s or hundreds or thousands of samples. So being able to kind of more, in an expanded view, be able to explore the lineage of any of those samples is critical. So back on the sample, well, so let's actually, from this particular sample, we could say that we wanted to create more derived samples. So we go through our create sample IDs, indicate which sample type it is, say that we're going to generate 20 more. We could say also as an input, we're including some other media or things of that nature. Add that as a parent as well. And then let's not make 20, let's just make 5 of these. And then when we hit finish, it creates these 5 new sample IDs. Now the way the sample ID I've configured it to just be the prefix of S, today's date, and then just an integer counter. So these are the first 5 samples I've made today. But all of these then also have that same lineage that we can go back and look at. I had also added, the mixture batch, the media recipe, as an input to this sample as well. All right, I'm going to go back to, so how do we kind of look at how how these samples are connected to assay data. So in this case, you can see we have a link to the assay data that was linked to this particular sample. And if I go click on that, it's going to take, well, I should click on the right thing. I want to click on the specific row to see what was the result connected to that particular sample. And you can see that we've got all of that amino acid analysis for this particular sample right here. And we can download that if we need to. The other thing to mention is you might have multiple assays that are connected to a particular sample. In that case, they would all show in that sample detail page. So now we're in assays. I just kind of want to show a little bit more about, in this case, we've only defined 3 different assay types for the system. If I go look and visit that amino acid data as well, we can look at the full results, and these are all of the different assay results that are tied to, that have had amino acid analysis done to it. And I can do searches and filters and all that type of manipulation in here, and I can also download information as need be. Finally, if I need to import data, we've got a mechanism for bringing in more data here as well, so that tied to that particular assay ID. And we can actually even see the template of what that data is supposed to look like. Now, obviously, you set this up once, but you can see we've got this Excel spreadsheet, in this case, shows us what the sample ID is supposed to, I'm sorry, what the assay information is supposed to look like. All right. So that's samples and assays. Now going to return to the PowerPoint. I want to talk just a little bit about media registration. So this is, it's been interesting. Half the folks we talk to love this, being able to track this. They want to be able to optimize bioreactor runs. Other folks, this is not of interest. So I'm going to show a little bit about it, but we are cognizant of some folks don't necessarily feel like they need this in their workflow. Other folks very much want to. It allows you to register media recipes in batches, track the specific lots of the raw materials used, and then for, connect to experimental results. And really that's through samples. So you're always able to see what are all the samples that are connected to this particular media batch. The other thing I want to, and I'll demo that in a sec, but workflow management. So this is really a tool to enable that team-based science, being able to track requests for samples, assays, anything you want to be able to request within the system. When you come into it, you see the open tasks that are assigned to you, so it helps an individual understand what tasks are on their plate. But you can also see the status of everything you've assigned that's still open. Maybe you need to go nudge someone to do that assay that you've requested. It connects then the request to the resulting data. So once someone has done that assay for you, there is a persistent link between the assay request itself and the data that was uploaded as a result of it. You can look at trends, and it also allows, for a manager to be able to see, okay, how many times have we measured titer? How many, instruments do we need? Are there bottlenecks either in personnel or instrumentation that we need to address to sort of improve the efficiency of the lab? So again, bouncing back over to the application itself. So here is our media registration module. And we have a variety of ingredients that have been registered. These are all kind of, and you can register these one by one, but obviously you want to be able to just upload an Excel spreadsheet or something like that that has the definition of all the ingredients that you are using within the lab. More important is the actual mixtures themselves. And so ingredients and mixtures are really the virtual definitions of those things. So a mixture here is just a recipe for how to make that media. And then each time you need to make a batch, you make you come to this particular recipe and hit create a mixture batch that allows you to actually make something that you're going to use in the lab. So we have information about these ingredients. This is not the most interesting of media recipes I recognize. I don't think anything would actually grow in it. You know, lots of cases where you have 40 or 50 different ingredients, and then down below you have the various steps for making that particular media recipe. From there, we can actually see that you can create a mixture batch. You can indicate a little bit of information about this, but then we have tools for the actual preparation. Say how much of the media that you want to make. In this case, we're going to make 20 kilograms, and then we have tools down below that tell you the amount desired, the amount you used. You indicate, you know, which raw material you used, and notes around its preparation. And then that is all saved within the system. All right, enough of media. The last kind of part of this module is the workflow module. Now, this is the view for someone who is using the system. In this case, I don't have any requests that have been assigned to me, but I have 3 that I've assigned to others, and it lets me know, you know, what type of request, what the title of that request is, and who it has been assigned to, so I can understand a little bit more about. You can, from this system, request obviously additional work. So we'll call this, you know, webinar demo. We can put information about that. We'll assign it initially to Josh D. But we want to keep other folks aware of this. We want to, in this case, connect it to a particular expression system. So you can see we have lots of kind of hooks in there that connect it to other data types within the system. And we could assign this, also make sure that we're including a very specific sample that is tied to, in this case, that sample. And then most critically, we can have different services, as many services as we want. So in this case, we'll do a sample request and a purification, assign that to Josh, and then follow that up with an assay request that's tied to SEC, that's tied to myself. We can put more information as we want around the description. So you have kind of a top-level description of what the work request is, but then the specific services down below. And this is all highly configurable. You can have as many types of services as you want for each one. You know, you might have very different types of sample requests within your system. And so that can be very much configured for your specific workflow. And then finally, we can attach files to this if it's a SOP or some sort of critical paper, so it's also a part of that system. Go to create it, and this is obviously a fairly simple one, but it is now, you can see now I've opened a couple of other requests, but assigned to me we have this very specific webinar demo SEC assay request. Okay, so we've gone very lightly over kind of the 5 different areas. I want to sort of zoom back though a little bit, so not zoom in, zoom out. And really, one of the key things I want to emphasize about LabKey Biologics, we have all this different functionality for managing a biological workflow, but key to LabKey Biologics is the ability to bring data in from a variety of different sources. So, you know, currently running assay instruments and inventory systems also down there would be already, if you already have some sort of registration system or pre-existing database of sequences, we make it easy to write that data into LabKey Biologics. Similarly, we have lots and lots of different mechanisms for pulling data out of LabKey Biologics into other analysis tools, other systems that need information that is going to be generated in there. So one of the strengths of LabKey, and it really isn't a new thing with LabKey Biologics, it really stems from that foundation of LabKey Server, is the ease in which you can bring data in and out of the system. The other thing is just sort of in terms of the development flexibility on top of it. Everything we've built in LabKey Biologics is API enabled. So you know, when we provide an API for registering protein sequences, it's the same API we're using within the application itself. We also have APIs in a variety of different languages. So there's a lot of, whatever, hopefully the flavor of development that you might do in your sort of scenario, we've got. This isn't for everybody, but for folks who do have developers and staff and want to build their own custom modules or specific hooks around scripting, we give you the mechanisms to do that. So, specific resources, and we'll share this PowerPoint with you. Again, my name is Ryan Luce. My email is here. Kevin Krause is the lead developer on LabKey Biologics. We have lots of public documentation on labkey.org about biologics. So you can either wait for this PowerPoint link, or you can go there directly. And there will be little a lot of the documentation. There's short videos showing a little bit more in depth of how different features work. And then we'll actually share the video from this particular webinar with you guys soon. But at this point, I'd really like to open it up to questions from folks that are here, so we can make sure to address, and we can jump back into the application if needed, questions that you guys might have that I can answer.
Thanks so much, Ryan. All right, if you do have a question, go ahead and enter that into the question panel. You can also chat that if that's easier for you. We're going to give everyone just a couple of minutes and see what kind of questions come through, and I will share those with Ryan. All right, Ryan, so the first question they have is it only a one-to-one relationship between assay results and the sample ID? For example, I might have an assay that runs the sample at multiple dilutions of replicates. How is that handled?
So it is absolutely not a one-to-one relationship. So first of all, you might have replicates, just like you said. So you might have so you can have for each one just different. Excuse me, I got a cough. And as you upload that assay data, it absolutely accepts that and will track that sort of appropriately. And if you do dilutions, we can also handle that. We can either sort of define the sample in terms of dilution, or we can add that as an attribute to the assay itself. So it very much does handle that.
All right, awesome. Thank you. Okay, I've got one more question here. If one already has an existing registration system or inventory system, does LabKey allow you to connect to the existing system with the proper API while keeping the rest of the features?
Yeah, yes. I mean, and I guess just to re-emphasize that, we don't come into any situation where there aren't kind of pre-existing situations, unless it's a brand new company. Even in that case, generally people start to populate some sort of database very quickly. And so, and one of the strengths of LabKey is just being able to leverage that, all of those various APIs so that you can directly connect from an existing registration system or whatever system it may be. So absolutely, yes, we can make those connections.
Okay, awesome. Thank you. All right, well, if you do have any additional questions that come up, please feel free to reach out to Ryan or to myself via email after the webinar, and I can route those to the correct person, make sure that those get answered for you. The session was recorded. I will be distributing that as soon as I can get it all cleaned up. And thank you again so much for joining us, and thank you, Ryan, for taking the time to present. So everyone have a great afternoon.
Yeah, thank you guys for attending. Much appreciated.